Hi, everyone. I would like to introduce Dr. Chan-Hyun from Johns Hopkins University School of Medicine. Dr. Na has worked in Senju about 10 years ago as a postfellow. He had extensive expertise in proteomics, including LC-MS, MS-specific mass spectrometry, imaging mass spectrometry, bioinformatics, and computer programming. Additionally, he has been trained in neurodegenerative diseases and molecular biology. His lab work focuses on systems biology and translational research, identifying the novel proteins and developing technology for the cell-type-specific proteome analysis to reveal pathogenesis mechanisms. Today, Dr. Na will present a seminar entitled In situ cell-type-specific proteomics analysis to study neurodegenerative diseases. Let's welcome him.
Thank you, Aijen. So, I'm really happy to come back to, Memphis in about 11 years. So then I was surprised that, Memphis has been changed a lot. So it looks much better, compared to last time. So yeah, when I walk around, it was very scary, but it looks like it's much less these days. So yeah, and also thank you for the invitation. This is really my honor to give a chance to talk, my study. So before I talk about, today's project, actually, my lab, focused on mass spec-based proteomics. So we apply to biomarker discovery for various types of neurodegeneration, and also we are expanding into many other diseases like sepsis or POTS, many other those kind of diseases, and also we use this mass spec technology to identify some non-canonical proteins, because although we are defining let's say there are about 70,000 proteins expressed in the human cell, but when we look at expressed protein from genes, actually there are a lot of alternative translation, maybe initiation site, and also there are many proteins coming from alternative ORFs. So we found many, currently more than 1,000 non-canonical proteins expressed in alternative frame or alternative initiation site. So, and another one is this kind of technology development for cell type specific proteomic analysis, and also in another branch we are doing mass spectrometry imaging based spatial metabolomics and proteomics, and also cell type specific metabolomics as well. So today I'm going to talk about cell type specific proteomic landscape of brains with neurodegenerative disease. Yeah, so I don't know how much you are familiar with mass spec, but whenever I give a talk about mass spec, then people didn't know about mass spec, much less know about mass spec than I expected. So I tried to give some background about mass spec. So, mass spec mostly LC-MS is composed of LC part and LC part and mass spec part. So once you have a peptide or protein prepared, then you separate them by hydrophobicity mostly. Then it is infused to mass spec. So this part is quite similar to HPLC or FPLC, whatever you are doing in commonly. But after it is separated, then it is directly infused into mass spec instead of collecting fractions. So then, and also it is in real time. I mean, once it is spread into, then real-time data is collected, like it is first of all precursor. Then peptides are infused into mass spec. Then it is fragment. Actually, it is detected in peptide level first. Then mass information is recorded. Then once mass information is recorded, then there are interested peptide you want to fragment. Then you infuse those peptides again. Then they will be isolated for your target peptide. Then it'll be fragmented here, then and push back to here, then goes to orbit trap, then identified here for their mass. So although there are multiple other ways you can analyze using this kind of mass spec, that's basic way of analysis. So then what happens is once when you infuse those peptide into mass spec, then real time it is collecting like these days even 150 or even a bigger number of spectra per second. Then once you run like maybe 2 hours of mass spec, then you'll have maybe have more than 100,000 spectra if you stack them like this thick book. You can make using the spectra. So then once you open those stacked spectra book, then it is composed of MS1 precursor mass. That means peptide precursor are detected. Then next page is composed of MS. That means fragment of those peaks. Then after you fragment all of them, then it goes back to MS1. That means precursor scanning. Then you do fragment of them. So this is just repetition of this one. So once you have MS2 spectra is collected, then you are comparing to theoretical spectra derived from human protein database. So because when you have a human protein sequence, then you can predict what will be generated peptide when they are digested by trypsin. Then you can generate the theoretical spectra from there. Then you can compare those theoretical spectra to actual spectra you acquired. Then you can find the best spectra, match your theoretical spectra. Then in this way, you can identify. Although there are a lot of statistical analysis and many other methods implemented here, but that's basic idea, how we are identifying proteins there. So then, many people are using this approach to identify proteins, differentially expressed or regulated in many samples, like animal sample or human sample. So common approach we are using is, for example, for Parkinson's disease, Parkinson's disease brain proteome analysis, we extract, let's say, human brain, there is substantia nigra, that's the human brain region, in which Parkinson's disease patients have neurodegeneration. So we extract brain tissue from PD and healthy control, then extract the protein from there. Then we can analyze them using TMT based proteomics technology. These days, many people also using label-free proteomics technology. So there are maybe many multiple different branches, but we used to hear TMT based proteomics technology in which you can multiplex sample at that time up to 11, but currently even it is can be multiplexable to 36 sample. Then anyway, after TMT labeling, at the time 11-plex was available, so we split those 30 samples into 3 different batches. Then to normalize between 3 different batches, we put common sample, reference sample. So based on this sample's intensity, we can normalize abundance data from 3 sets. Then each sample will be pre-fractionated for their peptide. Then LCMS analysis will be done. Then database search will be done for protein identification. Then we do a lot of statistical and bioinformatic analysis to figure out what kind of pathways are dysregulated. So then, after completing this experiment, we identified around 10,000 proteins. Then this is just, data normalization using combat package. So we did, statistical analysis. So this, volcano plot, I don't know how many people are familiar with this one. So protein on the right side is increased protein in PD, and left side there decreased protein in PD. And also we did statistical analysis in other way that is called bootstrap AUC analysis. So probably I'm not going to go to detail, but anyway, differentially expressed protein here, upregulated, downregulated, could be quite similar. So we found overlapping protein in two different approaches for differentially expressed protein. Then we used the DG protein for statistical analysis. So what we found was ribosomal proteins were a lot of ribosomal proteins were differentially expressed in the human brain of Parkinson's disease patients. So we did further analysis. So although we did many different analysis, but I'm just showing key point here. So what he found was rather than just ribosomal protein, actually mitochondrial ribosomal proteins were main proteins that were differentially expressed and regulated in Parkinson's disease brain. And also there are some RNA processing related proteins and mitochondrial electron transport chain related proteins were dysregulated as well. So then once we have this data, then you can say, oh, it looks like mitochondrial ribosome protein is really important in Parkinson's disease patient, but still we are missing, I think, critical information. Is that the only pathway, maybe the only important pathway dysregulated, and also then what cell type they are came from? So this is, we always have this kind of question. So for example, as you can see here, in the brain, there are, multiple different cell types exist, neuron, astrocyte, and microglia, and also oligodendrocyte in maybe endocellular cell, even neuron, there are multiple different type of neurons. Then, when we see, this kind of a change, then it could be, there are a lot of possibility. Maybe it can change the only one cell type or different cell type, or sometimes there could be many different pathway, proteins involved in pathway, they are upregulated here and downregulated here. Then when you combine them together, looks like there's no change. Then we can miss, very critical pathway. That's important for, to understand the pathogenesis mechanism. So then how we can resolve this issue? Definitely cell type specific, proteomic analysis could be a solution for this one. So many people have spent a lot of effort to understand, how those, how we can approach, develop the method to do cell type specific proteomics analysis. So, in case of Aaron Schumann group in Germany, they developed, Bonkem mouse that has mutant type of tRNA synthase is expressed in specific cell type using Curilox system. So in that mouse, when they have a mutant type tRNA synthase is expressed in certain cell type, then when you feed those mouse with ANL, that's methionine analog, then this ANL will be incorporated into protein in place of methionine. So then this ANL incorporated protein can be retrieved by click chemistry using and then labeled with biotin. Then you can retrieve biotin-related protein, then identify cell type specific proteins and figure out what kind of pathways are dysregulated in specific cell type. But problem is you need to do genetic modification, and also this one only applicable to newly synthesized protein. So and also it could be if you label them for a long time, it could have toxicity, and also number of identified protein could be quite less than 3,000 proteins. So then Srikant Ragaranju group now in Yale University, his group developed the actually turbo-ID based cell type specific protein method. They used quite similar approach as this one, but instead of cell type specifically expressing methionine tRNA synthase, they expressed the turbo-ID, that is biotin ligase. So once you have biotin ligase expressed in specific cell type, then you can once you feed the mouse with the biotin, then you can have proteins on target cell type biotin related. Then you can enrich those proteins in the same way as you did here. But this one also needs genetic modification, and also number of identified protein is quite limited, less than 3,000 protein, and also let me see, yeah, and also only maybe some cell type it works, but for some cell type it does not work well. So because of that, some people use laser capture microdissection. So once you have brain tissue section or other tissue section is prepared, then you can cut out each cells. Then you can do cell type specific programming analysis for them. But although we put tissue section, then it is still three-dimensional. So there could be other cell type on the top of that or bottom of that, or even though you cut out those target cell using laser capture microdissection, but there are a lot of contaminant protein coming together, and also even staining and cutting the target cell accurately is quite challenging. So some people even try fax-based cell type actually separation, but once you dissociate those cells, then you will lose a lot of protein information, and also protein information can change during sample preparation as well. So this one also is not quite often used. So then what's the solution? So then we have tried actually other different methods previously, but that also did not work well. Then what came to mind was, as long as we can have a certain enzyme that can modify protein, especially for biotinylation, then we can achieve a cell type specific programming analysis. So then we have already good method, immunohistochemistry. So for example, if you want to label proteins on astrocyte, then the astrocyte have a GFAP protein, expressed cell type specifically. So you can use anti-GFAP antibody. Then secondary antibody has, horseradish peroxidase. This is already enzyme. And also you can, if you want, you can put other enzyme. So then, what happens is when you do it, then people usually put chromogen, then they color them using this HRP-based activation of those chromogen. But if you put there inactive biotin tyramide, then this biotin tyramide, actually biotin tyramide is, you can just consider that this is kind of a biotin, but there is some functional group. So then, and also this is used in, this system is used in Apex-based interactome analysis. So once you put biotin tyramide, it will be activated by horseradish peroxidase. Then this activated biotin tyramide will label on tyrosine residue in the vicinity. That means protein on astrocyte. So you will have most of the astrocyte protein biotinylated. Then you can also color them to check their cell type specific biotinylation using streptavidin and HRP and biotin system. That's called ABC kit. Then those biotinylated protein can be retrieved by lysing the cell, tissue, then enriching biotinylated protein using streptavidin beads. So then you can identify those enriched protein using mass spec. Then what you identify here will be protein from those astrocytes. So in this way, we can achieve a cell type specific proteomic analysis. So advantage of this one is, you can apply whatever tissue, that's applicable for, they're subject to immunohistochemistry. So you can even apply to any animal model, any human tissue, and also you don't need any genetic modifications. So anyone who can do immunohistochemistry can do this cell type specific proteomic analysis, slightly modifying their immunohistochemistry protocol. Yes.
Other proteins a cell, or is this just specific to, let's say the antibody you are using. So you only interpret antibody, right?
Yeah.
You actually will be spread to all the other proteins in the cell.
All the other proteins in the cell. That's actually a good question. So this idea actually also used for interactome analysis. For example, if you are interested in GFAP, interactome, then you can use the same system, but if you control the level of biotinylation, then you can only make GFAP and interacting protein biotinylated. If you put more reaction time or higher concentration of biotinylation, you can make entire protein on the cell, biotinylated. Yeah. Yes.
Dissociation of the protein. So in a way, it's pulling down.
Yeah.
Biotin complex, right?
Right.
From the target or how you break down the cells, that may change what you pull down.
Oh, actually, no. We are completely lysing entire, cells and protein will be all dissociated into solution. We are using SDS, buffer. So SDS buffer basically, and also we are sonicating. So basically, all the cell will break down, and also protein will be dissociated, become soluble. So from there, we are just enriching biotinylated protein using streptavidin beads. So for this one, after we do cell type specific biotinylation, we have to break the tissue. Otherwise, if you just pull down tissue condition, then cells still they are connected to each other, so it's very difficult to pull down, separate. So we have to break it. But anyway, your target proteins are biotinylated already. So as long as you can enrich biotinylated protein, then you can achieve cell type specific proteomic analysis. Yeah. Let me get my water. Yeah.
So this method is heavily dependent on the specificity of the antibody.
Yes. Yes. So, but many
good antibodies are very rare. And
Oh.
they always have some background. So how do you sense that the threshold say,
Yeah.
this is the signal I want?
That's
the background.
good question. But to achieve cell type specific proteomic analysis, you don't necessarily need to achieve 100%. So, for example, you have 90% of biotin is on astrocyte, then maybe 10% or 5% on other cell type. Then still you are enriching, 90% of protein from astrocytes. So then, for example, there are certain protein included here and decreased here. So if you combine them together, you don't see differences. But when you enrich 90% from here, then you can see the difference. So, but in our experiment, but it's quite specific, but it's also depends on optimization. So, whatever tissue you are working on, for example, you are working with the brain, then there are mostly interested cell type. Then you already have, a cell type specific antibody. And if you are working on kidney or heart, you also have, specific cell type you are interested. Then they are doing, cell type specific staining, for their analysis, because they need to figure out cell type, right? To show, by immunofluorescence or whatever. So
You have like anatomical landmarks to identify, okay, this is the cell.
Yeah.
But once you lysis the tissue, then you don't have any positive identification.
Any what?
Like any positive.
Positive identification.
Yeah.
So, what do you mean by, so when you say landmark, they mean in structure, state, but once you have a biotinylation of your target cell type, then structure does not matter anymore. You can just break it and enrich biotinylated protein. But
once you break it,
yeah.
You don't know what we are looking at.
Oh, so you're saying spatial information will be lost? Is that what you're saying?
Basically, you are not looking at cells. You are just looking at some pieces of cells, and they can come from anywhere. From the tissue from.
No, so it is from target cell type. So actually, I don't know. Yeah, I don't know what part we are not in the common page. But for example, if I spray red color on your clothes, then even though you go to Memphis anywhere, I can collect you person. So then we can easily figure out, oh, they are from UT. Yeah.
In this method, because it's tissue section, do you get better recovery of like GPCRs, like membrane spanning proteins, like in the case of neurons?
Oh, membrane spanning protein. So it also depends on what antibody you are using. But actually, that's also a good point. We don't necessarily need to use one antibody. If you're interested in other different type of, especially you're interested in membrane, then you can use, maybe membrane-specific antibody. And also, probably, I don't want to go to detail, but because, we are also thinking about next generation of this technology. So we are using multiple different antibody. We can even achieve cell type specific subcellular organelle proteome, and cell type specific membrane proteome. Those kind of proteome analysis is possible. So we are actually designing that now. We are acquiring data. So okay, so then although I said just idea, but whether it is working really or not, that's critical part, right? So then we proved the cell type specific analysis in this way. So we prepared mouse brain from healthy mouse. Then we first one group is just no primary antibodies applied. Another group is no primary applied. Then another group is neuron specific antibody, and third group is astrocyte specific antibody. First group is microglia specific antibody. Then we did the proteome analysis for them. So although I said this, we combined that because we used the TMT-based method, but you don't need to maybe pay attention much. But anyway, we did proteome analysis for them. So before we are doing proteome analysis, we first of all looked at whether they are cell type specifically bioattenuated. So this color, we did the cell type specific bioattenuation, then we colored them bioattenuated cells using chromogen. Then as you can see here, this NUN, this is hippocampus, then hippocampus clearly, then dentate gyro, CA1, CA3 region, they are, bioattenuated. Then also astrocyte, that shows, typical astrocyte shape here, the staining. Then IBA1, this is for microglia, also it shows typical microglia shape, staining. So then we did proteome analysis using those tissue. So after we prepare sample, before we do enrichment, we analyze this sample first, whether there is differences. Theoretically, there shouldn't be any differences. And also, when we analyze this sample, there should be big differences because they are from different cell types. So what we observed was when we analyze the sample before enrichment, then there's no difference. When you compare astrocyte to microglia, no differences. And neuron to astrocyte, no differences. But when we compare enriched sample, then this astrocyte and microglia shows completely different expression pattern. That means those astrocyte specific proteins are are here. Then microglia specific proteins are here. So those proteins are probably quite common in two different cell types. So then GFAP is astrocyte marker protein is upregulated. This is another proof that it is cell type specific. Then also this is IBA1, protein. So it's also downregulated. It is same for comparison between other two pairs. So in this way, we proved that this is quite cell type specific. Then we are very excited. Then it is quite cell type specific. So then although we saw this marker, but are those proteins are really cell type specific? Then what we did was we used those highly expressed proteins in one cell type compared to other cell type. Then we did cell type analysis. So because single cell RNA-seq field, they already picked the cell type specific marker proteins. So using that database, we checked whether our those highly expressed protein in one cell type shows expected enrichment. So this neuron cell type specific protein data shows glutamatergic neuron was most enriched cell type. Then same for astrocytes, same for microglia. So this one proves that our enrichment works perfectly for our target cell type. So then, probably I think it's quite related to, questions you asked. Then although we did the cell type specific analysis, then is that only we are enriching, protein surrounding those GFAP or, target protein we used. So because this technology initially used for interactive analysis, so we analyzed whether they show overall even distribution over multiple different subset organelles. So this white one is non-enriched sample. Then this is GFAP, astrocytes, neuron, and microglia. They show quite similar pattern, except for this, neuron, because we used NUN marker. This is more focused on actually nucleus of neuron. That's the reason there are a little more protein biased toward the nucleus, but overall they show quite similar pattern to non-enriched proteome. So, yeah. Yes. Right.
If you get the 15%, 20% plasma membrane related protein, these are the kind of cells?
No, not outside. They are from inside. They are mostly inside. Yeah. Because although we did tissue section, but all the membranes are removed using acetone or other molecules. So it is already penetratable. So right. So we tested that even looks like it's working for sections with about 150 micron thickness. So it is quite working well for very thick sections. So when you do it, you don't need to make it thin section, just make it thick, or even just chop into smaller pieces. Still, it will work. Yeah. So then finally, we applied this technology to 5xFAD mouse model. This is well-known AD mouse model in which human amyloid precursor protein is overexpressed. So then, because we just used the common well-known mouse model to see whether it shows a very typical expected result or quite different results. So we used the wild type and 5xFAD. Then they are labeled for each cell type, like neuron, astrocytes, microglia in the same way. Then in the same way, we enriched those proteins, from each cell type, then analyzed by mass spec. So before doing proteome analysis, we checked whether examined their cell type specific bioaccumulation we could achieve. So because some people concerned that in 5xFAD especially, then those astrocytes or microglia, they are activated. Then there will be more maybe staining. But as you can see here, it didn't show clear. Very big differences. It's quite similar. So GFAP, did not show much differences. IBA1 did not show much difference. Although cell shape is a little more simpler. But this is already known that when they are activated, then their cell shape is there are less branch actually. It's 12 months. Yeah, actually, yeah. This
know that the microglia are activated.
Right. Yeah. So
why are you not detecting that?
Why we are not detecting that?
Why are you not seeing increased EBA signal?
Oh, so yeah, actually that part we are not sure. This is what we see. So we didn't see much difference in.
That's not fact. That's a very, very well established.
You are saying more, but it's not more number, right? There are more number of microglia.
Yes.
Okay, so maybe I should look at more carefully. But anyway, this is what result we got.
Increased around like 3 or 4 months. By 12 months, it's a huge increase.
Okay. Yeah. So maybe I may need to look at more carefully whether this is we are capturing all the microglia or we are missing some. So anyway, this is proteome analysis result. So this is conventional proteome analysis. That means it's not cell type specific. So they are increased protein and those are decreased protein. So out of about 8,000 protein, then 1,500 proteins were differentially expressed. So this is quite similar result to other publication in which they did cell type protein. I mean not cell type, just bulk proteome analysis for 5X FAD mouse. So then we were surprised by the cell type specific protein data. Neuron proteome data, about 3,000 proteins out of 8,000 proteins were differentially expressed. Then astrocyte is similar. About 3,000 protein, microglia, maybe slightly smaller number, but still much bigger number than this one. Even you can see the number is quite different, from what you are seeing here. Then what kind of proteins are differential, cell type specifically? I think this is almost world first data we are presenting in 5X FAD for cell type specific. So, and also nobody have achieved this depth. So then, in conventional proteomics, mostly immune activation, related proteins are prominent. So maybe I need to explain a little more of this data. So this is just enriched pathway from this differentially expressed protein using those differentially expressed protein. Then this most enriched, then second enriched, the third, fourth enriched. Then this one shows that this first pathway, what kind of, whether they are increased or decreased. So when it is red color, this is more increased. Then also this one also tells the individual proteins whether they are increased or decreased. So mostly they moved into increased direction for those pathways. Then we went to individual cell types. For neuron, surprisingly, mRNA processing pathway that was never observed here was the most enriched pathway for neuron. And also Dr. Junmin Peng in St. Jude, he's studying this one, AD brain mRNA processing really important. We also found this one here. So then cytoplasmic, ribosomal protein and calcium regulation related to proteins. So then, those mRNA processing proteins are quite upregulated. And also this second one also quite upregulated. But third one is downregulated, fourth one downregulated. So they are showing, quite, unique patterns and directions. And astrocytes, they also show quite different patterns from neuron. So calcium regulation was most enriched pathway in astrocyte and G protein signaling pathway. So this is also quite interesting because G protein signaling, to my knowledge, is important for tripartite junction in which neuron to neuron, the synapse junction, and there are astrocytes, also involved there. So that junction, G protein signaling pathway, important in astrocyte. So this is dysregulated in astrocyte in the 5XF8 mouse. And EGFR1 signaling pathway in microglia is quite similar pattern we've seen here. Pathogen phagocytosis pathway, IL-1/5 signaling, and thyroid, causal network in microglia. So this is not, unexpected thing. So then we did weighted gene co-expression network analysis to delve into further because, when we just look at those differentially expressed protein, then, there are pathways, then we don't know whether they are coming from really same pathway or they are, multiple proteins from multiple pathways are, combined together, then that's misleading. So that's the reason we use the WCNA, because I don't know how many people are familiar with WCNA. WCNA is, kind of deconvoluting proteins based on their expression pattern. For example, you have let's say 20 samples across that, columns. Then certain proteins show, maybe this kind of expression pattern. Certain protein will have this kind of expression pattern. Certain protein will have this kind of expression pattern. Then based on this expression pattern, we are clustering those proteins, because protein that shows similar expression pattern are considered probably coming from the same pathway or similar pathway. That's the reason we did the WCNA. Then those modules are, you can consider them protein that shows, similar expression pattern. In other words, coming from the same pathways. So then, this red color mean increased in 5xFAD, and blue color mean decreased in 5xFAD. And size of a circle means significance. So the bigger significance, the bigger size it is. So then this M11 module was increased in 5xFAD. Then this M5 module was decreased in 5xFAD. Then we also compared it, known AD markers and their correlations. For example, probably this also, I need to explain them. So there are a lot of markers known involved in AD, or also there are a lot of proteins that work as a marker protein for those cell types. This is, this data is from single cell RNA-seq data. So we compared their correlation of those protein in these modules with the marker proteins. So then, but when you look at them, there's no clear pattern for, the correlation with those neuronal cells when you analyze this bulk proteomics. But when you analyze those neuron-specific proteome, pattern is completely different. So this M1 module was decreased in 5xFAD. This M9 module was increased in 5xFAD. But as you can see here, most those neuron-specific markers are correlated with this one. That means those protein, excitatory neuronal proteins, we can think those excitatory neuronal proteins are downregulated So that's the interpretation we can get. Then this is more related to Fragile X syndrome pathway. So that's the information we could got from here. Then when we analyze astrocyte, then this M11 module proteins were increased in 5xFAD, and M17 modules are decreased in 5xFAD. Then when you look at this correlation with the marker, then here, protoplasmic astrocyte proteins are highly correlated with increased protein in 5xFAD. Then fibrous astrocyte marker proteins are highly correlated with decreased protein in 5xFAD. That means probably I'm a little confusing. So fibrous astrocytes, probably this is more enriched in white matter. This is more enriched in gray matter, or it could be reversed. But anyway, their pattern of expression is quite different. So then when it comes to microglia, it's quite expected. So maybe there's nothing special, so I'm going to skip this part. Then we further, look into how they are. What about the subcellular compartment, proteome? How they are looking? So then, as you can see here, in case of nucleus, it's very difficult to read here. So I'm gonna read it here. So the spliceosome protein was, mostly upregulated in neuron, nucleus. Then, on the other hand, those nucleus protein was, spliceosome protein was downregulated in the nucleus of astrocyte. So that means when you analyze them together, then nucleus protein and spliceosome related protein, nucleus of astrocyte downregulated, increased here. So when you combine them together, looks like no change. That's the reason we are missing very important information here when you do bulk proteomics. So this is very representative example. We definitely need to do cell type specific proteomic analysis. So, and also in the mitochondria for neuron, a lot of oxidative phosphorylation pathways are downregulated. So then in the mitochondria of astrocyte, also, they are downregulated for oxidative phosphorylation. In that case, we can find it. So I'm not gonna go all the details because we are running out of time. But as you can see here, they are opposite direction, same direction, pathways. So definitely we need to do cell type specific proteomic analysis. So then there are a lot of Alzheimer's disease related marker proteins were found, so for example, Emory group, they found that expression pattern of Alzheimer's disease related pathway was different depending on their disease status. For example, this final stage is, let's say your symptom already, patient already show symptom. Then this is category 1, like this is appearing like about 30 years ago, then 20 years, 10 years, something like that. So then that means amyloid plaque and material from changing occur first. But it's already, not surprisingly, this is A-beta overexpression mouse model. So A-beta is already overexpressed in neuron, astrocyte, microglia. And metrosome, probably this is overexpressed, but I'm gonna skip this part. But synapse change related protein, although they defined they are synapse related protein, but surprisingly they are downregulated in the astrocyte instead of neurons. So because they thought maybe this is more related to neuron, but actually it's changing in astrocyte. But one thing I'm careful is this is 5xFAD mouse model. So just A-beta overexpression mouse model, but this data is from human, human sample. So maybe there could be difference, but there are some information still we can get it. So glycolytic metabolism mostly downregulated in astrocytes. And if you go to synaptic loss, synaptic and neuronal loss, expectedly downregulated in neuron, but also some of the protein was downregulated in astrocyte. And functional decline related protein actually did not change in neuron, or more changed in astrocyte, upregulated there. So this one also emphasizes that cell type specific proteomic analysis will help us to better understand what's happening in the brain during pathogenesis process. So, and also, I already told you there are a lot of proteins showing different pattern between cell types or only expressed in one cell type. This is, exemplary proteins. So those proteins only changed in neurons. So those proteins are upregulated only in neuron, or this protein is downregulated only in neuron. So, and also it's same for astrocyte. There are a lot of protein changing only in astrocyte, a lot of protein changing in microglia. But when you combine them together, probably neuron only takes about maybe 10%, less, about 10%, or could be higher, but 10% of entire protein in the brain, then it's diluted. So it is not statistically significant anymore. That's the reason you are missing those kind of proteins. And there are a lot of proteins that are showing opposite direction. In that case, definitely you will miss those proteins, when you combine them together. This one, further emphasize why cell type specific proteomic analysis is really important. So then, next question is, okay, you showed that, mouse data, then you said it will be applicable to human. Then does it really work? Human could be different. So that's the reason we did the same way of analysis for human brain. So we did biotinylation for neuron, astrocyte, microglia. Then this one shows biotinylation, almost similar pattern what you seen in mouse brain. So then we did actually statistical analysis in the same way as we did for mouse brain. So this is before enrichment. This is after enrichment. So as you can see here, there was complete difference between enrichment and after enrichment, showing it is cell type specific. And also, we did the cell type enrichment analysis using those differentially expressed protein. Then, as expected, the neuron specific, astrocyte specific, microglia specific was observed for neuron, astrocyte, microglia proteome. So then, probably in time restriction, I'm not gonna go detail this one, but we also did midbrain of Parkinson's disease mouse model, but we also proved that this is working really well. But maybe the exciting part of this one is, we are using very small portion of brain. So protein amount is quite limited, but still it worked really well for those Parkinson's disease mouse model. So I'm gonna skip this part. Then, then what about other cell types? You said, neuron, astrocyte, microglia, it is working for them. Then does it work for oligodendrocyte, or what about endothelial cell, other cell types? So then we tried actually 3 different antibodies. So MOG is more like, broad staining of oligodendrocyte, even including myelin-based protein. So that's the reason, overall bioaccumulation you can see here, covering a lot of exons. Then SOX10, this one mostly stay in cell body of oligodendrocyte. That's the reason you are seeing only circular shape. And oligo2, this one mostly staying in nucleus of actually nucleus and also oligodendrocyte precursor cell nucleus. So that's the reason you see much smaller staining, the size of staining. Then what about other neurons like cerebellum? There are different type of neurons, so like a granular cell or Purkinje. Does it work? So that's the reason we did the proteome analysis for Purkinje cell Purkinje cell in neuron and also granular cell. Purkinje cell in the cerebellum and granular cell in cerebellum. And also we did the astrocyte microglia too. So as you can see here, then Purkinje cell, it shows clear cell type specific staining pattern. Then granular cell, the same. Then astrocyte, sorry, this is not neuron, this is microglia. Also, it shows clear staining. So this is preliminary data. Just we compared whether they show expected pattern. Then yes, it showed expected pattern for Purkinje and granular cells because marker proteins are expected and changed in an expected way. Then what about other neuronal cell types? We just showed only NUM-based staining, but there are a lot of different types of neurons, like excitatory neuron, inhibitory neuron, even inhibitory neurons. There are parvalbumin, GABAergic neuron. There are many different types of neuron. Then can you do that? So because of the we did analysis. So parvalbumin staining, it works, and also serotonergic neuron, it works. Somatostatin neuron, it works for that. And GABAergic neuron, also it works. And glutamatergic neuron, it works. Although there are some background actually, as you indicated. So but we may need to maybe improve, but anyway, it is working. Then cholinergic neuron, it just works for it. So yeah, and also endothelial cell, I didn't include it here. It is working as well. So this is a team of my team who involved in there. So Dr. Tae Gam, he's also associate professor at Johns Hopkins. He provided mouse model, and Dr. Tae-Kyung Ryu, he's postdoc in my lab. And he was made actually critical contribution to development of this one. Initially, this method did not work well, but once he started working on this, then it worked really well. So Dr. Seokyeong Kim also involved in this project, and Dr. Juan Tromposo and Dr. Javier Redding Ochoa, they provided a human brain, and also other people involved there. So this study was funded by NIH and Harris Foundation. Thank you. Yeah, I think people already asked the question. Yeah, please.
Thank you for the lecture. It was excellent. I liked it a lot.
you.
You took samples from midbrains.
Yeah.
Normally, when they have like a neurodegenerative disease, for example, depression, everything, I read some articles that they put electrodes like surgical on in the cortex.
Mhm.
They try to activate the cortex. Means more impact is on cortex or midbrain because of neurodegenerative. Because
I didn't catch the last part. Say that again.
means, for example, in the cortex, for depression, if somebody is having severe depression, they go for surgical, like placing electrodes in the
Yeah.
cortex for stimulation. Even those medicines like SSRI, they act in the cortex more.
Mhm.
So much more. Why means that's why I want to know about that. Why samples were from midbrain and cortex? Do they differ in neurodegenerative?
So the 5X FAD, we used to actually brain covering from cortex to striatum, all the area. But for midbrain, I thought you are talking about this Parkinson mouse model. In case of Parkinson mouse model, we use midbrain because that's the brain region. Neuronal cells dies by those alpha-synuclein injection. The first, but although after all, it dies, it kills other neuronal cells as well. Yes.
I remember in one slide you showed that the MAP protein is decrease in astrocytes, but not in neurons. So when I saw, I was scratching.
You said
my head.
MET protein?
MAP protein. So that's predominantly a neuronal protein.
Uh-huh. Oh, so yeah. So although there are predominantly neuronal protein, but still, there are most protein they are expressed in other cell types. So one of the example was probably
And A beta is predominantly expressed in neurons.
yeah, but
Not in glia.
yeah, but in this case, this is A beta overexpression mouse model. So this is overexpressed everywhere.
Mostly in neurons.
So, but A beta, you're saying A beta, but although it is mostly neuron, still it is expressed in other cell type, right? So it is not.
So the main
So-
pathology.
Yeah.
But you are detecting the minor things. So the two, MAP is mostly a neuronal protein,
Mm-hmm.
and the A beta is mostly in neurons,
Yeah.
not in glia. But you are somehow detecting this minor.
You are saying contamination from other cell type?
If I see it, if I'm doing it, I will think about that.
Yeah, but the protein can be expressed in other cell type. So
Certainly that's.
just abundance is different, but still it is expressed there. So it is not surprising when we detect those expressed proteins.
Pathology is mainly in neurons.
Yeah, so
And you are not seeing the main pathological consequences.
actually I'm probably not understanding what you're asking. So yeah, that is important in neurons. So we are catching what's happening in neuron when A beta is overexpressed, but the A beta is also overexpressed in other cell type as well in the mouse model we used.
Yes, but it's mostly in neurons.
Yeah, so that's the reason we
And
analyzed
the
it.
most of the map is mostly in neurons. So the two main things, it's causing nothing, but some minor changes are being picked up. So I'm just curious, why is that? Certainly that's possible, but I'm just curious.
Minor changes picked up. So we are separating proteins, depending on their cell type, then we are catching changes. So then if I were, reviewer of the paper, then when the author detected that minor protein in the cell type, I already know it's expressed there and minor, then if it's detected, then this method is really, really sensitive. So still, I don't understand why that become problem.
Makes sense after there. Something like mine. Okay, that's just my curious. I'm
Is
just
it
curious.
possible due to the normalizations issue, like you normalize, right? So theoretically, it has to be, let's say, the GFAP levels, N levels, and the IGA1 levels have to be equal among, say, the group, right? But then when the levels are different, let's say, the GFAP is lower or upper in mouse model, let's say.
Oh, yeah, you're right. Probably I think because of he's confused, I guess. Yeah.
So it's more because.
Yeah, right. So yeah, although let's say neuron has this expression level and astrocyte this expression level for let's say MAP, then when there is no differences, it is not shown as differential expressed protein. But in astrocyte, when you show this, although expression level is low, but when there's difference between groups, comparison group, then it is shown as differential expressed protein. I guess that's the part there's confusion, I guess. Yeah.
How do you normalize? How do you set the normalizing? Let's say like the animal models or even humans, the expression levels for, capturing, like proteins for capturing are different, right? So how do you normalize? Your data show, like say, UN versus GFAP or
Yeah.
granules versus QJ. But this is based on the mind that all the levels are equal. among the different subjects, right?
Oh, so I think you are talking about normalization between different cell types. They have probably different protein abundance in the brain, right? So yeah, we are assuming they have same protein abundance. So for example, neuron is more, there are more population of neuron, then astrocytes smaller. Then when you extract the protein abundance could be different. So, but we normalize them as if they have same expression level, then find differentially expressed proteins.
Yeah, like for other techniques, we always have like say housekeeping genes that do not change.
Yeah.
That perhaps the groups or the subject.
Yeah,
right?
yeah.
But of course, you made preliminary data saying that those levels are equal, but there may be a difference, right? How do you normalize it?
So I showed two different types of volcano plot. One is comparing between cell type. In that case, we normalized assuming they have the same expression level between two different cell types. But another data set I showed was comparison between disease and control group. In that case, we are comparing the same cell type in two different groups. So in that case, we don't need to assume they have same expression level. I mean, we don't need to normalize much. They are already showing similar expression level.
Way to validate your the data showing the difference in the expression levels are actually valid. Like let's say comparing it to messenger RNAs or some controls to say that this is valid.
Yeah,
like,
probably. I think if we do immunofluorescence, probably we can validate those expression level. Once we find target, then we can do immunofluorescence. Then without normalization, we can see how much they are different. Even between cell types and different research. I mean comparison groups. Yeah. Yeah.
It is actually, for example, the total RNA-seq, we are go up, let's say 9,000 or 8,000, 9,000 transcriptome. When we use the single cell RNA-seq, we only detect maybe 2,000, 3,000 gene expression. Of course, I mean, in your model, you have the limitation. Probably, I mean, you can detect, my question actually, how many different proteins you can detect in your mass spec? For example, we have 20,000 genes. We are expecting 20,000 proteins. This is the first part. And also, in your method, can you also analyze the alternative spliced version? I mean,
Yeah.
first part, actually, how many different protein you can detect in a given cell type? And then the second one, actually, I mean,
yeah,
can you detect the alternative spliced
yeah.
version?
So we are detecting around more than mostly 8,000 proteins, but depending on what mass spec we are using, then how many samples we use, then we can even go up to 10,000, even higher. So this is one of the strengths of our technology compared to other methods. Then alternative splicing, that's actually also a good question. So maybe there are a lot of alternative, splicing differences between cell types as well. But in that case, we have to go even deeper proteome analysis, because when we say we identify the protein, when we identify only one peptide from the protein, we say we identified, then we are comparing abundances. But they probably can come from different isoforms, right? In that case, it's not fair comparison. So, but we are not reaching that level yet for low abundance protein. High abundance protein we can do that, but low abundance protein we are not doing that. So in that case, we have to do even deeper proteome analysis. Maybe larger protein amount could be required, or very sensitive state of art mass spec. Not like Orbitrap Astral or timsTOF Ultra 2 could be the solution for that. Yeah.
I have a question about your technology. I think this is very interesting. Is there an alternative to biotin that you can use?
Oh, yeah. So that's also, we are trying to do that in the lab. So maybe you can use the histidine tech can be used. So, and also, as long as you can enrich them, you can use whatever, tech like, HA tag or MVP, whatever. So theoretically it's possible, as long as it's not too big. So if it's too big, it's difficult to handle because of aggregation or other things. But if it is small enough, then you can use the tech. So we are already using those kind of differential tech method for like cell type specific subset of organelle or cell type specific membrane proteome, those kind of things we are developing now.
One last question.
Yeah.
I just have a confusion. I want to clarify. Means when neurodegenerative disease occurs, it is more impacting midbrain or cortex, or and the second one is that from where they start mainly, from cortex or from midbrain?
So yeah, that's a good question. So it depends on the disease type. So if it is Alzheimer's disease, then it is known that it is starting from maybe entorhinal cortex region, then hippocampus, then other region, and also it depends on what molecule you are looking at. A beta and tau, they are spreading past different. So and Parkinson, although we are saying substantia nigra region is shows degeneration, but according to Brack hypothesis, that people think that it could coming from maybe gut to the brain or olfactory bulb to the those midbrain. So that's the reason we in this study we used that gut PFF, I mean alpha synuclein injected the mouse mouse model. So it migrated to the brain. Then we saw the those pathology. And also some people are trying to inject that to the olfactory bulb, then migrate to the brain. So it depends on disease type actually. Yeah. Okay. Yeah. Looks like there's no question. Thanks so much.
you.
Yeah.
Human models, one of my friends did some research. He found that
Would you come closer because people are moving. Yeah.
He found that some proteins get deposited in the brain that contributes to Alzheimer's.
Oh, acid blocker.
Acid blockers, like esomeprazole, omeprazole.
Yeah. So
He said that when you take this one, your Alzheimer's disease increases because they deposit some proteins in the brain.
yeah. So this was confidential, but one of my collaborators, they are studying about the pH involvement of pH in the neurodegeneration. Also, I can tell the detail, but there's I think.
This is,
Yeah, yeah.
I did not believe in it, but now you are saying that it may be definitely impacting
yeah.
because these
pH
PPIs,
is important.
because pH is important.
Yeah.
pH is important for the proteins.
Yeah, yeah.
Well, yeah, because, okay, yeah. Yeah, so we'll go out and.
Excellent,
Thank you.
impressive.